understanding better. We start the recording now, and the topic for the day is symptoms, complete reanalysis based on cognitive neuroscience, neuroanatomy, connectonomics, and genetics. Who else better than Bhaskar can throw us some light on this topic? Bhaskar, good evening. The floor is yours.
Let's remind people that we have started. Just let me. Just again ask everyone. Hello.
Yes, Bhaskar, you're audible. Bhaskar?
Hello.
Yeah.
Yes, sir.
You're audible.
Temporarily connection was.
Acha.
Am I not visible?
You're not visible as yet. You have closed your video.
No. Presently.
Your video is stopped.
Am I? Okay. Let me change the connection. I'm restarting.
Start your video.
Am I audible now?
Yeah.
Yes.
You are.
And visible? And visible?
Yes, you are.
Okay. So, are we waiting for more people? Because there is only 33 people.
I think you start, let people join.
Okay. So, first, let me apologize for slight delay and also today it won't be very nice to hear this topic till the last 5 slides. When you see the last 5 slides, you would understand what we are trying to do. But before that, you would be bombarded with a lot of molecular jargons and a lot of other things. I would try to make it simpler. I would try to describe as much as I possible. I would just request you to clarify whenever you don't understand anything. And with this forewarning, let's start the presentation. I hope with this presentation, you would have a basic idea of whole of psychiatry. So today we are going to talk about molecular psychiatry and molecular psychopharmacology and how negative and cognitive symptoms arise in psychosis and mania, and by discussing that we would automatically discuss how molecular pathophysiology of mania occurs. And I hope, although I am not going to describe them explicitly, that you would find molecular pathogenesis of a lot of other things in this presentation too. I am stopping my video so that we get the full bandwidth. So first, let's recap.
Sir, sorry
The,
for the interruption. One black box coming on your slide left corner.
yes,
Can you please drag it to open it? Yes, go ahead. Thank you.
already done. Yes. So this is what we discussed in our positive symptom formation. The delusion is formed due to increased autobiographical processes. Increased autobiographical process separately lead to rumination and obsession, and high or extended motivational reward circuitron separately lead to compulsive impulsive behavior, and decreased inhibitory control and decreased executive function separately lead to attention deficit trait, addiction to process and substances. But when these three combine, they give rise to fixed firm beliefs not amenable to logical reasoning, which is known as delusion. Now, the hallucination process is same. Delusion forming machinery is there. Along with there is disturbed sensory discrimination network, disturbed sensory integration network, which together would lead to disturbed spatial perception and orientation and decreased 3D placement of hours, and ultimately it would lead to hallucinations of all sensory modalities, though auditory is generally primary in psychosis. So this is the connectomic based formulation of delusion and hallucination. Then we have to think about the molecular mechanism, because connectomic formulation is just a bioelectrical field based formulation for the pathology we are dealing with. The real pathological things that are happening, they are happening at molecular level. And there are millions of causes and trillions of complex combination of these causes which can give rise to this type of network disturbances. So we need to have a workable molecular understanding that would make these millions and trillions of combination into a configurable molecular mechanism which we can address, which we do address with our drugs. So the crafts of molecular mechanism should be heightened neural, glial, and immune system hyperexcitability. Although there is one another process that frequently is mistaken and overlooked, but we will talk about it later. And the hyperexcitability, when it happens, it would then translate into individual or group of neuronal network causing deviant functioning, and depending on how and where the hyperexcitability is happening. So to understand the hyperexcitability, we have to first go into basic formulation and basic molecular understanding. The basic understanding of neural excitement during increased functional demand of brain would be our first journey. This is just a very simple flowchart which says that when there is increased homeostatic load, by increased homeostatic load, we are saying that when brain needs to work more to accommodate some change in either internal environment or external environment. So that would cause high information carrying load for brain because brain primarily carries information. This would lead to hyperactivity of brain areas handling the load. Sometimes it is sensory load, sometimes it is information load, sometimes it is somatic sensation load, sometimes it is motor planning and other type of load. So depending on which information brain is carrying more, the different areas that are handling the load would go into hyperactivity. That would lead to increased cellular protein recycling fast. Now, increased cellular protein recycling means for this process, there has to be a electrical and metabolic coupling. So electrical and metabolic coupling only can happen when there is high level of transcriptomic activity at the neuronal synapses, at the neuronal cell body, and all the areas of neuron that are working extra. That this would lead to more and more protein production, and during production, as there is rapid transcriptomic activity, many of these proteins would not reach their desired post-translational modification and their desired three-dimensional structure. Depending on cellular bioenergetics and depending on specific characters of these proteins, this would lead to increased destruction of these proteins too. And ultimately, that would create a kind of deviant proteostatic response, which would lead to cellular stress and ER stress. We will talk about it more details in next slides. Now, there would be increased mitochondrial oxidative phosphorylation to handle the metabolic demand. Whenever there is increased mitochondrial oxidative phosphorylation, there would be a lot of mitochondrial fission, fusion, and recycling, and there would be a lot of free radical production, which would lead to redoxes and cellular hyperactivation. Again, cellular hyperactivation would lead to cellular stress. Now, cellular redox stress is defined by various reactive oxygen species, reactive nitrogen species entering into cellular cytosol, and from the cellular cytosol they trigger various cellular activation. Now, depending on the cell's ability to counter this reactive species, initially there would be cellular activation, later there would be hyperactivation, and there would be various other responses in the cellular cytosol and membrane lipid, which would lead to cellular stress, and it would be sensed by NLR, nod-like receptor, RIG-like receptor, and various other pattern recognition receptor, and there will be resultant activation of DMP pathway. Now, we can make it a better flowchart here. Whenever there is increased cellular protein recycling, it would do three things. There would be increased production of proteins and high transcription and higher need of protein folding. Now, whenever there is protein folding, there is a unique group of protein that don't have either fully, they lack the three-dimensional structure, and they are called intrinsically disordered protein, or they have some areas of polypeptide which don't have a stable structure. Then that is called protein with intrinsically disordered region. These are the proteins that have a high level of action in cell. Various cells, various cell proteins have intrinsically disordered region, or they themselves are completely intrinsically disordered. By not having a rigid three-dimensional structure, they exchange stability for high functionality and mobility. They are able to do multiple works at once. Uh, Tumor suppressor protein P53 is one of the typical example of a protein with intrinsically disordered region, and by having that intrinsically disordered region, it helps in multiple ways to suppress the malignancy transformation and to suppress the various genetic defects from propagating into malignancy. Now, when the intrinsically disordered protein are produced more and more in terms of protein demand, there is higher chance of them getting misfolded. And that misfolded protein response would cause destabilization of microtubular, cytoskeletal, and cellular transport. That would lead to increased cellular membrane permeability and cell-to-cell transmission via gap junction, and that would lead to cellular hyperexcitation and cellular stress. Then there is another pathway, that is when there is increased cellular protein recycling, there will be increased activation of ubiquitin proteasome system, which is actually proteasomes. There will be increased activation of proteasome system, which is ubiquitin dependent as well as independent. That would lead to increased energy demand of cell and increased amino acid production and turnover. That would lead to increased reactive oxygen species production and oxidative stress. They are picked up by intercellular stress signaling pattern recognition receptor, and damp signal are activated. That is again leading to cellular hyperexcitation and cellular stress. Now, endoplasmic reticulum stress is sitting at the middle of it. It is activated by cellular protein recycling. It is activated by increased protein production, and it leads to more misfolding. It leads to cellular hyperactivation. It leads to more stress signaling recognition, and it leads to increased reactive oxygen species and reactive nitrogen species production. Now, whenever there is increased cellular energy demand, there will be increased mitochondrial oxidative phosphorylation, increased mitochondrial membrane permeability, increased mitochondrial dysfunction. That would lead to increased mitochondrial fission and fusion. That it leads to increased mitochondrial autophagy, which is also known as mitophagy. Now there will be increased mitochondrial synthesis to manage bioenergetics, and every process would lead to increased reactive oxygen species production, reactive nitrogen species production, would lead to oxidative stress, would cause widespread cellular excitability, and that would lead to cellular hyperexcitation and cellular stress. Now you think that these are actually biophysics, biochemistry talks. So why you need to know it? Because these are the pathways through which our diseases occur. This is mitochondrial fission and fusion. and their defects are common in psychosis, in autism, in ADHD, and in every other known serious mental disorder. Similarly, reactive oxygen species and reactive nitrogen species are responsible for microglial activation, astrocyte activation, then increased permeability of blood-brain barrier, lymphocyte infiltration, and ultimately neuronal injury, neuronal death, misfolded protein response, protein accumulation, mitochondrial dysfunction, and ultimately lead to every kind of neuroprogression, neurodegeneration, and neurodevelopmental disease. This is very short how the intersection occurs. Impaired mitochondrial function would lead to mitochondrial unfolded protein response. ER stress would lead to chaperone-mediated unfolded and misfolded protein response, and that would lead to high level of autophagy, which might be due to or chaperone-mediated autophagy or macroautophagy or microautophagy, and ultimately all these are dysregulated in all our disorders. So when we talk about misfolded protein response and we talk about intrinsically disordered protein, many of you are starting to think, what is intrinsically disordered protein? So I would go slightly deeper into this because this is one of the huge link between neurodevelopmental to neurodegeneration pathway. That doesn't mean, contrary to popular belief, that every neurodevelopmental disorder is going to end up neurodegenerative disorder. If biology was so simple, we wouldn't have been sitting here and discussing this. Anyway, so this is a folded protein, has a stable three-dimensional structure. This is a folded protein with these areas which are not really folded and they are intrinsically destabilized. And this is a fully disordered protein where there is no structure, no stable structure in the protein. P53 is a protein which has two stable regions and rest of the regions are intrinsically disordered. The intrinsically disordered proteins pervade and permeate every signaling pathway, be it autocrine, be it endocrine, be it paracrine, be it juxtacrine, all depend on intrinsically disordered protein. In fact, all the hormonal receptors, all the steroid receptors, all the receptors that you commonly talk about, means glutamate receptor, NMDA, AMPA, and serotonin receptor, and various growth factor receptor, all have intrinsically disordered protein fully as a component, or they have a intrinsically disordered region attached to a stable structure. Because without the intrinsically disordered region, no receptor would be able to do multiple function at a time. And intrinsically disordered protein also allowed tuning of signal responses. When binding to a protein, intrinsically disordered proteins actually can create fuzzy complexes or create an order upon binding, or there is a unstructured yet static conformation when they bind to the partner protein. All three are important in pathogenesis of our disorders. These are the various ways intrinsically disordered protein work in cellular function. I am not going into details of this because this is not for memorization, this is just for understanding. And if you look here, you would find all the major cell cycle pathways, they have actually intrinsically disordered response. This is one typical example of a receptor with intrinsically disordered region. For those who don't understand what it is NMDA receptor. This intrinsically disordered tail leads to multiple things done by NMDA receptor. Depending on various ligand binding and other things, this would lead to calmodulin kinase activation. This would lead to magnesium activated kinase activation. This would lead to calcineurin and various other pathway activation. And if the intrinsically disordered regions are not working, then NMDA receptor won't be able to do long-term potentiation or long-term depression. Then there are various other intrinsically disordered areas which are important in our disease. For example, the mammalian circadian clock, which determines the seasonality of psychosis and mania, have actually an intrinsically disordered protein directing it. So once we end with intrinsically disordered protein and excitotoxicity, we need to go to the next topic, which is equally interesting. I don't know how many you are finding it interesting, but it is very interesting because all of this would make sense when you go to the last 5 slides. That I can promise you. So anyway, now we are talking about human endogenous retroviral element, HERV. These are actually part of non-coding DNA. Non-coding DNA is 98.5% of DNA. We know that. And many a time, those who know me hear me talk that this is where most of our disease lies. That is true because this is where the disease causing mechanisms are lying. One of them is human endogenous retroviral element. Human endogenous retroviral element is nothing but DNA sequences within human genome that originated from ancient retroviral infection of germline cells. Nowadays, we are finding that the virus need not to be retrovirus. Various other virus also can bind to uh, genome and germline cells. But that knowledge is just 1 or 2 years. Now, these are involved in a lot of cellular processes and mostly control all the immune system responses in various ways. All our immune system responses, including various diseases of brain, are controlled by this human endogenous retroviral element, be it multiple sclerosis, be it other thing. And if any of you remember, 3 to 4 years ago, there was a huge hue and cry in nature that Neanderthal contributed many of human diseases. There actually the topic was human Neanderthals have given us many of these hu- uh, human endogenous retroviral elements. Now, HERV is closely related with schizophrenia. In fact, if you search HERV and schizophrenia, you would find increasing number of papers are being published on this matter. Again, this is a very simplistic view of what is HERV. First, one single virus affected the index animal, which might be a chimp or even something that is common ancestor to all the mammals. Then all the offspring would contain this specific retrovirus. Then a second retrovirus infection would occur, and third, they would then integrate with the first, and ultimately we would get multiple endogenous retroviral family. At this moment, we have roughly known that there are few hundred endogenous retroviral families. Now, how they actually cause various psychiatric symptoms? There is perinatal infection, which can be influenza or Toxoplasma gondii. Then there would be secondary infection. There might be or might not be. Even secondary infection, many a time not needed, which would be CMV or HSV1 or HSV2. That would lead to activation of HERVW, which is specifically targeting toll-like receptor 4. And there would be neutralizing antibodies to that, which would lead to pro-inflammatory condition and neurotoxicity, and ultimately that would lead to a subgroup of schizophrenia and a subgroup of bipolar disorder. This is another picture showing various human endogenous retrovirus and the superfamilies. And the way they actually cause various diseases, they either lead to molecular mimicry, which is the most common mechanism. Then they act as super antigen. Many times they lead to loss of epigenetic control. Many times they cause continuous inflammation, and in association with exogenous viruses, they can trigger all of these pathological processes. These are the top 15 diseases that has been found to be associated with HERV and the various families of HERV. The number is the number of paper published on them standing in 2024. Multiple sclerosis has 37 paper. Breast cancer has 20 paper. HIV has 16 paper. Schizophrenia has 15 paper. It is the fourth in number. So you see This is one of the highly researched and high topic in today's academic era. And this is the statistics of 2024. Now, this is one of the articles amongst these 15, which has shown that this a group of HRV protein causes deficit in glutamate synapse maturation and causes various behavior associated with psychosis. For those who want to know, this is HRV type W. Now, this is another recent paper which is discussing about evolutionary recent retrotransposon contribute to schizophrenia, and recent retrotransposons means the recent infection. The infection is which has integrated to germ cell, the more chance of it to become associated with various brain disorders, specifically schizophrenia and multiple sclerosis. Schizophrenia and multiple sclerosis both are not diseases, just symptom, but still. Then there is another paper which has shown that there is a subgroup of schizophrenia and bipolar patient who has HERVW antigenemia, and that can be detected by unsupervised cluster analysis of their various protein profile. So we have shown you the various ways through which pathological cerebral excitability occurs. Well enough, we have reached the pathological cerebral excitability. But what next? Because excitability means a lot of things. Even migraine is excitability. That doesn't mean every patient of migraine is going to end up as patient of psychosis or patient of mania. So we are missing something more, and that we discuss again later. But anyway, so we are talking about excitability. So we have talked about the way brains reach excited state during a state of increased function. So fundamentally, dysfunction is any of these pathways would lead to increased cerebral excitability and pathological excitability. The dysfunction can be increased working in this pathway or decreased working in counterbalancing pathway. But decreased working in counterbalancing pathway is very less evident so far. And ultimately, this would lead to increased excitability of cell in every part of body and brain if it is a germline affective process, or increased excitability of a part of an organ. In our case, it is brain if it is somatic cell affecting process. So somatic mutation or inherited mutation along with epigenetic changes. But as I was talking about, that doesn't make sense. Only hyperexcitability doesn't cut the cheese. There are something missing that we normally don't talk about, and that is the missing elephant. The missing elephant is neuronal migration and its dysfunction. The first universal disruption in brain. It has to be there, otherwise there won't be any psychosis, any mania. On it, excitotoxicity would be superimposed. But without neuronal migration dysfunction and neuronal migration disorder, there is no chance of any psychosis or mania happening. What is neuronal migration? During brain development, neurons are born in specific areas and need to migrate to their final destination to form complex circuitry of the brain. The migration process is absolutely essential for proper development and function of nervous system. The migration is of two types, actually more than two types, but these are the major types. Radial migration, where neurons origin from subventricular space and go along the glial pathway radially towards brain surface and ultimately settle down in gray matter areas. And that is the major form of neuronal migration happens during neurodevelopment in utero. Tangential migration is by following the brain's length where cells from hippocampal region, hippocampal progenitor niche area or subventricular progenitor niche area, cells migrate along the brain's folds and go to olfactory bulb and various other areas which are rapidly recycling areas. This is less prominent mode of transmission, but these two are main mode. There are other various forms of migration. I am not going into that detail and making you more bored than already you are, because I know most of you are thinking why we are even attending this lecture which is full of basic science thing. But I'm again saying, just let me go to the last 5 slides. Your patience should be rewarded. So the mechanism involved. Guidance cues. Guidance cues means these are proteins and mostly they are proteins of coagulation system, mostly they are proteins of various immune system which make those neurons and very the neuronal cells either push from one area to another area or pull towards one area. Guidance cue can be pull or push. Pull is mostly enhancing chemotaxis. Push is mostly repelling by hydrostatic charges or various other electrical charges. This means this is various amino acid synthesis system or the various inborn errors of metabolism gets involved and complement C4 gets involved. You see, you are getting slowly rewarded for your patient. We talked about complement C4 once, disrupted in schizophrenia gene and disc one once. They are actually Q proteins. This Q is actually function of complement system and all those things. Then various cellular adhesion molecules. Various cellular adhesion molecules enhance the chemotaxis, enhance the slow migration, and all the schizophrenia genetic consortium which are finding new and new hits in their genome-wide association studies, they are finding various cell adhesion molecules and their genes in the gene pool. This is where they come into. The various cell adhesion molecules contribute the migration process. Then cytoskeletal structures. If you go into details of psychosis genetic consortium finding, you would find a lot of actin mutation, myosin mutation, actomyosin complex mutation, how actomyosin movement happen that has a detailed pathway. Many genes of the pathway are highlighted in psychosis consortium and bipolar psychosis and bipolar genetic consortium findings. This is where they come into picture. Cytoskeletal structures give the slow movement to neuronal cell and they maintain the stability. Then intercellular communication and signaling. Those who talk about genetics of psychosis, they talk about renin protein and they talk about various growth factor promoter. They talk about various other things. This is where these two are the areas where reeling signaling comes. These two are the areas where various schizophrenia classifying protein comes. These are the areas where all the signaling pathways come. If we talk about schizophrenia and its genetic consortium, most of the genes are in this area. And intracellular signaling pathway, WNT, MAPK, all of them are heavily implied in psychosis. This is the area they come. So, this is how neurons migrate from progenitor area to their ultimate destination. And each of the stage is important for various dysfunction that affects this area and gives rise to formation of past vulnerability in every psychiatric diseases. Then what type of neuronal migration problem occurs in psychosis? There would be microscopic abnormality, molecular level abnormality, and these would happen during intrauterine period. This is where maternal fetal interaction comes into play too. Whenever there is maternal immune system hyperactivation, that would lead to various hits in this system, epigenetic hits, and the innate vulnerability is always there. So what is the connectomic result of this? The number of neuronal connection needed between hubs and node to function adequately go down. When we talked about connectomics and when we talk about various areas of connectomics, we talk about hubs and nodes. Hubs means large areas of brain where there is neuronal density and they control major functions. Insula is a hub. Anterior cingulate cortex is a hub. Basal ganglia is a hub. Rather, basal ganglia is a conglomeration of hub. Nucleus tractus serratus is a hub. Locus coeruleus is a hub. So what are the nodes? Nodes are the small neuronal centers that connect with these hubs. Hubs don't change, but depending on how our brain is trained and how our brain works, which areas of life we work more, depending on this, the nodes change. Now, when there is migration problem, the number of neural connections needed between hubs and node to function adequately go down. The resultant functional deficit causes the brain areas affected by deficit to stay hyperactive all the time. And this result in less than adequate functional activity and less than adequate functional achievement during peak demand. This would result in multiple deficit in brain performance throughout the life. Means, again I am saying, less than adequate functional connectivity and functional achievement. So, in these brains, there is not at first glutamate excess or glutamate mediated excitotoxicity. Initially, there is low signaling, low excitement. Initially, there is less than adequate glutamate level. Later, I am just giving example of glutamate. You can take it any other form. Later, if there is triggered excitotoxicity, then slight hyper excitation would occur that would trigger more and more excitation and more functional deficit, and ultimately it would lead to more date of the existing low connection and the connection deficit would increase. So this stall idea of glutamate excess, glutamate excitotoxicity in psychosis is wrong. Ideally, in psychosis, it would start with lower glutamate concentration, lower glutamate availability. And when we start finding glutamate results by various enema studies, we are sure to get low glutamate levels first rather than high glutamate. So, if we understand this, then we would understand two processes are there. First, migration abnormality that starts in utero and that continues throughout the life leads to low functioning of various areas due to less number of neuronal connection available between them, and they go into spurts of excitotoxicity when there is vulnerability in form of chance of developing excitotoxicity more and more. So first hit is migration deficit, second hit is various forms of excitotoxicity generation. Psychosis and mania occurs multiple hit ways, and migration also is not a single hit. I have shown you the multiple pathways of migration and how many millions of molecules are involved in that. So based on this, let's formulate what various phenotypes we are going to see in persons carrying these disorders or person carrying these vulnerabilities, whatever we like to call them. These are the pathological phenotype. Phenotype A. Here the neuronal migration disorder is severe and excitotoxicity is also severe. I have not mentioned mitochondrial disorders here, but you can understand that mitochondrial disorders would cause all these things. So I have not mentioned mitochondrial disorders separately. But mitochondrial disorder would result in severe excitotoxicity. So in phenotype A, there would be neuronal migration disorder in form of, let's say, some inborn error of metabolism. Along with that, there is excitotoxicity due to mitochondrial disorder. I am giving just example of two diseases that are associated with migration problem, severe migration problem, and severe excitotoxicity. So when they combine, the phenotype would develop seizure, multiple physical abnormality, intellectual retardation, extreme compulsive behavior, extreme self-harm behavior, extreme various forms of self-injurious behavior, and ultimately they would die due to sudden cardiac or epileptic phenomena. Then there would be a phenotype B. Migration problem would be more than excitotoxicity, but none would be mild. They start as autism spectrum symptom with anger discontrol, violence, impulsivity, and extreme compulsive behavior. Let us slowly or rapidly develop psychosis with or without seizure and social withdrawal. They end up as childhood or a very early adolescence onto schizophrenia, and we pile them on clozapine. Then there is phenotype C, predominantly migration disorder with slight excitotoxicity. Severe withdrawn autism spectrum symptom with gradual progression to old level simple schizophrenia. This is what simple schizophrenia is. And that we don't see simple schizophrenia nowadays because we diagnose autism spectrum more. So we don't let them go into that level. Then there is phenotype D, less migration problem and more excitotoxicity. They start as Asperger or high functioning autism spectrum disorder and gradually increases violent behavior and demands and end up as either UPD or cluster B or bipolar with borderline or chronic mania with obsessive compulsive symptom status. They are labeled in various way, but ultimately all of these goes around in this way. Excitotoxicity more, migration less. So the excitotoxicity is initially seen, later the migration problem come around. Phenotype E, mild migration problem and severe excitotoxicity end up as fast attention deficit, then bipolar, and ultimately when migration problems start manifesting themselves more and more due to progression, neuroprogression, they get seizure affective type. Phenotype F, most of the patients follow in phenotype F actually. Migration and excitotoxicity load is mild. Variable mixture expression leading to social misfit, eccentric drag, and who, depending on a lot of epigenetic vulnerabilities, would end up in adult onset depression with psychosis, then would go into some form of initial mania. They would be tagged as hyperthymic, they would be tagged as brooding, they would be tagged as a lot of things. And so with this, I end my complicated presentation. Now, I am hoping that I have given you enough thoughts, and I hope you would start asking me questions.
Thank you, Bhaskar. That was very comprehensive and very insightful. There are no questions as of now in the chat box, but I would like to ask you a question. How let's start discussion. So that will probably
No. The first question is how to treat. That is next. That is next one.
okay.
That
Yeah.
is next one.
Yeah. Okay. So yes, when we were talking, Dr. Divya Mangla has asked a question that yes, as you mentioned, how do you treat? So, depending on, let's say the last 2 or 3 slides that you showed us, the different type of phenotypical presentations depending on the mixture of migration as well as excitation. How would how would
That
you?
would that we would discuss in details over 1 hour in next one.
Yeah, but that brings me to the question is that let's say we have a phenotype, any of the phenotypes that you discussed about. Uh, in that phenotype, whatever are the presentations of the disorders. I mean, different people are going to have a variable kind of a presentation.
Exactly.
So, does a particular treatment, a particular drug, or any other a modality of treatment work for all in the same kind of a phenotypical subgroup?
No, no, they cannot, because this is a very gross phenotype. Now, when we analyze this, I have actually combined millions of causes into this 6 phenotype. They are not even stable phenotype.
Yes.
If we go into BSNP data, we would see that initially they created 3 groups: biotype 1, biotype 2, biotype 3.
That's the
Later in B,
BSNP's findings.
but even in BSNPs, they are now claiming after the BSNP 2 results are out that biotype 3 is not stable, and that is degenerating into either 1 or 2. So
Okay.
BSNP
Yeah. So
will
before we get into more of BSNPs, can you just tell us what this BSNPs is and what are the findings in very simple words?
yes,
Because people also have to know that when you talk of formulations like this, there are also some studies which have started kind of validating the type of concept that you are talking about. So can you just give a
yes,
brief of the BSNPs study?
yes. BSNPs was a serendipity, to be very exact. BSNP was a trial which is known as bipolar schizophrenia network of intermediate phenotype. They were trying to find a validation of the RDoC model by finding some biomarker that would validate the diagnosis of schizophrenia or bipolar. What they did is they got a large number of patients in 6 centers distributed all over USA. In these centers, they recruited patients, any patient who has psychotic bipolar, who has schizoaffective tag, who has schizophrenia tag. So they were trying to find the psychosis continuum. Ultimately, when they got enough number of patients, they mixed all the patients up, and they then applied a lot of biomarkers, which are mainly neuroradiology and evoke potential based biomarker, on these groups. They wanted to bring 3 groups of people out of the mix. One would be schizophrenia, one would be psychotic bipolar, and one would be schizoaffective. And they would come with their own biomarker profile. But this is where the whole thing backfired. When the 3 profile came, they indeed got 3 profile. But the 3 biomarker profile, when they came, they didn't came as schizophrenia. They didn't came as schizoaffective. They didn't came as bipolar. Rather, they created 3 stable aggregates where some people were schizophrenic by diagnosis, some people had bipolar by diagnosis, some people had schizoaffective by diagnosis, but in biomarker profile, they fitted one single group, and they didn't know how to do with this. So they gave the data name of biotype 1, biotype 2, biotype 3. And they started publishing the data starting from 2013. So when they get this kind of data, which disrupted and challenged their whole nation of schizophrenia and bipolar difference, they actually started following up these patients. And that created the BSNP 2 trial. They they wanted to follow these patients up for 5 years. Now, by biomarker profile, they created biotype 1, biotype 2, biotype 3. Biotype 1 is very severe. Biotype 2 is not severe but severe enough. Biotype 3 is mild. Initially, they thought that these three are three different kind of diseases that they found, and they proclaimed them more stable than DSM diagnoses. But when they started following these things up, they realized that this is not even a three homogeneous group. These are three heterogeneous group based described by level of progression of underlying pathology. So biotype 1 remains stable. Biotype 3 started degenerating into 2. One is going towards biotype 2, one is going towards biotype 1. And ultimately, they are now saying at the end of BSNP 2 that biotype 3 is not a stable biotype, rather, it is the early stage of disease. And they are even saying that if long-term follow-up is done, probably uh, all patients would ultimately degenerate into the most severe category, that is type 1. But this last part, they are not yet sure yet because they have not followed these patients for 15 or 20 years. This is just 5-year outcome.
Okay. So, yeah, that's quite something. I'll try and put up the links for this BSNPs papers 1 and 2. Let me see. Okay, now Dr. Rajesh Kumar has asked a couple of questions, and I'll take them up. So we talked about this IDPs, right? So Dr. Rajesh Kumar asked why the structural neuronal migration and functional IDP not happening in animals. Yeah. So I construe this from absence of schizophrenia animal model. So I think, see, this kind of proteins are going to be present in each and every cell, but the phenotypical presentations in a disordered state would be much different from, I mean, between animals and human beings. So we may not have a schizophrenia model, or I mean, of what happens in human beings is going to be the same as that happens in other mammals or lower animals. It might be something else. But this kind of proteins and this kind of mechanisms are going to be present in each and every cell of each and every multicellular organism, or maybe a unicellular organism also, for that matter.
Yes.
Another question he asks is IDPs also result of poor building blocks, macro or micronutrient deficient diets in successive generations?
No. Intrinsically disordered protein are actually essential for our body function, because let's say we have 20,000 genes. So we have only 20,000 copies of a protein. Many of the genes produce actually a single protein, and many of the genes are not going to produce a specific a lot of copy of the genes in every cell. So ultimately we have, although almost few billion proteins in a cell, most of them are various forms of or various duplication of a single protein. So if we have such low level of proteins, low variation of proteins, then we need to use them very efficiently. And to increase that efficiency, intrinsically disordered proteins have come into evolution because these proteins lack a single structure. They can adapt to multiple structure. Sometimes, for example, let's say I am again giving example of P53 because P53 is almost known by all of you. P53 is a tumor suppressor protein, right? That is one function. P53 is a nuclear surveillance protein and P53 is a DNA surveillance protein. That is number two. P53 is a protein that is involved in programmed cell death. That is number three function. P53 is a protein that decides when various organelle, that is number five or number four, I already forgotten the number. And if we go on listing the P53 work, you would find that P53 does more than 20 different functions. It is all because of that intrinsically of multiple configuration, and multiple configuration is an evolutionary advantage. But that evolutionary advantage gets too much problematic when cell is rapidly synthesizing protein and rapidly destroying protein. That time what would happen is intrinsically destabilized protein would go into various conformational change, and many of these conformational change would trigger various protein destruction mechanism because they don't fall within a particular bioenergetic parameter. Our cell is very tightly regulated bioenergetically and structural diversically. If a range of structure diversity is crossed or a range of bioenergetic signature is crossed, then the protein would be tagged for destruction. And that is what is happening when there is rapid protein synthesis and destruction. Intrinsically destabilized protein are getting misfolded and creating misfolded protein response. And so it is a double-edged sword.
Okay. So basically what we are trying to say is that these proteins, because of their not rigid structure, I mean, see, when we use the term intrinsically disorganized or disordered, it implies that it is abnormal to start with. It's an abnormal protein. But basically it means that this is a protein which, because of its flexible structure, can help other proteins, can help other cellular processes by kind of attaching to physically or in some other way. But at the same time, given if it becomes more structurally deformed, it can lead to degenerative changes or other processes like apoptosis. This is what I've understood by the concept of IDPs in health and disease. Okay, fine. So Dr. Prashant Chaudhary asks two questions. Yes, since there is so much importance of mitochondrial dysfunction, can we say that maternal inheritance holds more significance in psychiatry? One. And second, can psychiatric disorders themselves lead to disorders of energy metabolism in the body? So one is whether maternal inheritance is more important, and second, whether the disorder itself leads to mitochondrial dysfunction secondarily.
First question would have been differently answered if we were talking about this in 2018. Before 2018, I would have said yes, maternal inheritance is very much important, and maternal inheritance is a very closely associated thing with for psychiatric problems. But 2018 was a watershed year. That year, there were almost 5 papers which showed clearly that not only maternal mitochondria, rather paternal mitochondria also enters a zygote, and many of the time paternal mitochondria stays in zygote. They are not destroyed. So that creates an important area in mitochondrial medicine. There is now proof of biparental inheritance of mitochondria. Question is, we have not yet tried this for human babies. Means in human babies, we have not yet observed parental inheritance, but in various animal species, it has already been found. So it holds to the reason that in humans too, biparental inheritance of mitochondria is true. So this is, yes, before 2018, maternal inheritance would have been important. Now we should say paternal and maternal inheritance both are important. Now, can psychiatrists themselves? Yes, they can, because the thing is, when I was discussing, I was always telling about progression. The way things stand today, everything has a chance to progress. How much we don't know. Some would be progressing then static, some would be progressing at a very slow stage, some would be progressing then stopping, then again progressing, then again stopping, and sometimes by going to a particular checkpoint would stop. Some would rapidly go on and on till that patient dies. So standing there, yes, having mitochondrial problem in the root of various psychiatric problem, there is a chance that after disease manifestation, or rather after symptom manifestation, that would lead to more metabolic problem.
Okay, fine. So I think that brings us to the end of the questions. But okay, Dr. Paresh asks, how can we explain different types of schizophrenia and bipolar 1, 2 by this, whatever we have discussed today?
Dr. Shah, the question is, we have already destroyed schizophrenia and bipolar by this presentation. This presentation clearly says that there is no schizophrenia, there is no bipolar, that is just a given name by us. That this presentation's last 2 slides has clearly shown how the tags are generated, and there is no bipolar 1, there is no bipolar 2. Pakistan should rot in hell for creating these types.
Okay. Right. So, right. Now, Dr. Deva Mangala asks again, how can we explain post-viral fever, depression, and psychosis, psychotic symptoms in a few patients?
They are people who are carrying mild vulnerabilities. They had previously some deficit. Then after viral fever has happened, there is temporary drop in brain function due to high immune activation, and that has led to overt expression of this trait, which is seen as depression, and sometimes it is seen as psychosis.
Okay. Dr. Irene Rusdi asks, in what way does understanding this underlying mechanism help us to choose medication for our treatment? It's basically then the question that that was the first one to come. But I think we should make an yeah. So I think we should make an attempt to answer this question, because all said and done, we need to we need to be making a constant effort to apply our understanding so that our patients get treated better. Yeah. So how do we go about
we are going to arrange the next immediate next seminar on that. We would be discussing how this understanding translates into our treatment of our patient. We would be validating various old treatment. We would be finding new ways of treating that. I personally don't want to go into that because that would create a very long discussion and I would surely miss my train because that would take at least 1 hour.
Okay, fine. Uh, Dr. Usman Hotiana asks, so what can we expect in future to be destroyed effectively? So I think the first thing that we can expect is that we need to get rid of the concept of just a symptom-based uh, diagnosis and symptom-based treatment, because we are just kind of doing a very superficial job at that. We need to understand the processes better so that not only we can target our treatments better, but at least this can give us better leads into what we need to look at in the future. What basic research needs to be done? What kind of things do we expect? Since we have a lot of good technology available. I mean, when we talk of B-SNPs, there are so many things. There are EEG markers and MRI and this and that, including genome-wide association studies.
Yes.
Everything was there. So, well, that is what we should be looking at. Okay, again, one question: how substances like cannabis induce psychosis?
Cannabis use is always done by people who has underlying vulnerability and cannabis repetitive use. I am saying, I am not saying one of recreational use. One of recreational use can be done by anyone, but repetitive cannabis compulsive use can only be done by a person who already has vulnerability associated in the brain, which causes them to have less attention span, which causes them to have more anxiety, more brooding, and more other thing. So you are seeing that we are already looking at a picture of attention deficit trait, mild to moderate, obsessive trait, more prominent. So on this brain, cannabis gets associated. Now, with cannabis, there is actually not a single chemical. We are talking about 500 different chemical or more than that. Many of them would have vulnerability towards THC and problem in their endocannabinoid pathway. This in these areas, THC would cause activation of endocannabinoid pathways, cannabinoid receptor 1, cannabinoid receptor 2, various other pathways, and ultimately that would lead to suppression of endocannabinoid tone by repetitive use of THC. And the endocannabinoid tone is actually initially it increases inflammation, but later on constant use it actually suppresses inflammation. Now inflammation is also a name of growth. Without growth signaling and inflammatory signaling are same. When growth signaling gets excess, it becomes known as inflammatory signaling. So by doing the suppression of inflammatory signaling, chronic cannabis use is leading to less growth, more exacerbation of vulnerability, and ultimately it is triggering the psychosis.
Okay. I think, as you mentioned, this can go on and on, because once we understand this basic kind of processes, uh, how they affect the trajectory of the disorder, right from in utero to in utero over the lifespan, and how do how can we make attempts to intervene and see what we can do about it. So thank you so much, Bhaskar, for that nice discussion. And thank you to everyone for taking time out on Wednesday to take part in this discussion and uh, making it worth our while to present topics like this, because this also helps us to understand what we have read and what we have understood better. With that, I hand over the proceedings to Dr. Rajkumar. Thank you, sir.
Thank you, Dr. Malay and Dr. Bhaskar. Thank you all, friends. Thank Alchem for the logistics. Good night to all of you. See you again after a fortnight.
Thank you.
Bye-bye.
So the links are there. I put it on the chat, 4 or 5 papers. So those interested can go through this set of papers.
Yeah.
Thank you so much.